Welcome back to the Deep Dive. Our mission, as always, is to take the latest, often pretty dense research and break it down, give you the essential knowledge you need quickly and hopefully entertainingly. Today we are diving deep into a disease that's, well, it's rapidly become a global epidemic. It's called metabolic dysfunction-associated steatotic liver disease, MASLD for short.
Bit of a mouthful.
It is. You probably know it better by its old name, fatty liver disease.
And we really need to stress the scale here. This isn't some niche problem. Our sources are clear. MASLD is the most widespread liver disorder on the planet.
Really? How widespread?
We're talking roughly 1 in 3 adults globally.
Wow. 1 in 3.
Yeah, think about that. 1 in 3 people walking around might have this. And often, especially early on, they have absolutely no idea.
Okay, so what's actually happening inside the body with MASLD, and why is it a bigger deal than just, a liver issue?
Well, fundamentally, it starts with fat building up inside liver cells. Steatosis, that's the technical term. And over time, that buildup can lead to inflammation, scarring, eventually serious damage like cirrhosis or even liver failure.
Right.
But here's the crucial point, the one that really connects to what we're discussing today. It's strongly linked to cardiovascular risk.
Ah, so not just the liver.
Not just the liver at all. MASLD patients actually have a significantly higher risk of dying, but often it's from heart attacks or strokes, not liver failure itself. It's really a systemic metabolic problem.
Which makes finding effective treatments incredibly urgent, especially if it affects, like you said, a third of the population.
Exactly.
But drug development for liver diseases, this one in particular, hasn't had the best track record, has it?
No, it's been frustrating. Developing a totally new drug for MASLD is incredibly expensive and time-consuming.
And risky.
Very risky. Most compounds that look good in the lab, well, they end up failing in clinical trials. Often it's safety concerns. The liver is sensitive, and giving a drug long-term to people who might feel fine requires an extremely high safety bar.
So we need a smarter approach. Which brings us nicely to the core of today's deep dive. This strategy called drug repurposing.
Yes.
And we're looking specifically at 2 existing drugs, Pemafibrate and Telmisartan, that researchers from the University of Barcelona think could be a powerful combination for MASLD.
Drug repurposing is exactly what it sounds like. You take drugs already approved for something else and see if they work for a new condition.
Take a shortcut.
A massive shortcut, yeah. These drugs already have a known safety profile in humans. They've been used. People know the side effects. They're generally acceptable. So you skip years of early safety testing.
Which makes it faster, cheaper.
And arguably safer, because you're starting with known quantities.
And the researchers specifically targeted the early stages of MASLD with this strategy, right?
Wow.
Why focus there?
Because the goal is prevention. In those early stages, people usually feel fine. They're asymptomatic.
Right, they don't know they have it.
Exactly. But that's the critical window. That's when you want to intervene to stop that fat buildup from progressing to inflammation, scarring, fibrosis, cirrhosis, all the bad stuff.
And if you're treating someone who feels healthy, safety is paramount.
Absolutely. Safety is number 1. Which makes repurposing, with its known safety profiles, a really attractive starting point for these early stages.
Okay, let's dig into the 2 specific drugs the team, led by Marta Alegret, paired up. First, there's Pemafibrate. What's that one?
Pemafibrate. It's a lipid-lowering drug used mainly in Japan right now for hyperlipidemia.
Which is just high fats in the blood.
Yep, high cholesterol, high triglycerides. So it's already designed to deal with fat metabolism.
Okay, makes sense. And the other one is Telmisartan.
Telmisartan. That's an antihypertensive, so a very common drug used worldwide to treat high blood pressure, hypertension.
Okay, so one tackles fats, one tackles blood pressure. When you put it like that, the logic for combining them for MASLD patients, it kind of clicks.
It really does, especially when you remember that cardiovascular issues are the main driver of mortality in MASLD.
Right.
And what are 2 of the biggest risk factors these patients almost always have? High blood pressure and high blood fats.
So this combination isn't just aimed at the liver.
Exactly. It's hitting those systemic risk factors, too. It's a dual-pronged attack, really.
A 2 birds, 1 stone kind of approach.
Pretty much.
But the researchers suspected it might be even better than that, didn't they? They thought combining them might have synergistic effects.
Yes, that was a key hypothesis. Combination therapy is often explored because, well, first, you're hitting the disease from multiple angles. Pemafibrate works on lipids, Telmisartan works on pressure and maybe inflammation.
So you get broader coverage.
Potentially a more powerful effect, yes. But there's another huge potential benefit, especially relevant here.
Which is?
Dosage. If the drugs work together synergistically, you might be able to use a lower dose of each one compared to using either drug alone at full strength.
And lower doses mean fewer side effects. Back to the safety point.
Precisely. Lower dose, potentially less toxicity, better safety profile for long-term use. That's critical for a chronic condition like MASLD, where people might be on medication for decades.
Okay, so the theory makes sense. How did the Barcelona team actually test this? What models did they use in this study?
They started with a pretty standard approach, a rat model. They fed the rats a diet high in fat and fructose to induce MASLD, mimicking how it often develops in humans.
Right.
But they also used another model, which is quite interesting, zebrafish larvae.
Zebrafish. Why zebrafish for liver disease?
Yeah, they're becoming quite popular in metabolic research. They're simpler, cheaper to work with, and you can test things much faster, high throughput.
But are they relevant? Is their metabolism similar enough?
Surprisingly, yes. Their basic carbohydrate and lipid metabolism and even their liver structure and function share enough similarities with mammals to give really useful initial data. It's a smart way to screen things more efficiently.
Okay, strategy, repurposing, targets, fat and pressure, using Pemafibrate and Telmisartan, models, rats and zebrafish. Now, for the results, what did they actually find? Did the combo work?
Yes, the main finding was a clear success. The combination therapy significantly reversed the fat accumulation in the liver caused by that diet.
So it cleaned up the liver fat.
It did. It helped return the liver to a healthier state in these models.
That's great on its own, but you mentioned synergy and dosage. Did that pan out?
It absolutely did. And this, I think, is the really standout finding for potential patient impact.
Okay.
They found that giving half a dose of Pemafibrate combined with half a dose of Telmisartan.
Half doses of each.
Yes, half of the standard dose for each drug used alone. That combination was just as effective at reducing liver fat as giving a full dose of either Pemafibrate or Telmisartan by itself.
Wow. So half plus half equals the same effect as a full dose of one.
Exactly. That's huge.
That really underlines the safety benefit, doesn't it? You're getting the same liver benefit with potentially half the systemic drug exposure.
Precisely. Think about treating millions of people long-term. Reducing the required dose by 50% without losing efficacy. That's potentially revolutionary for managing side effects and long-term safety. It shifts the whole risk-benefit calculation.
And were the benefits just confined to the liver? Or did they see those wider cardiovascular effects too?
They saw those too, because you're using drugs that inherently target cardiovascular risk factors.
The lipid-lowering one and the blood pressure one.
Right. The combination therapy also lowered blood pressure and improved cholesterol profiles in the animals.
It's like a triple win.
It really is. Less liver fat, lower blood pressure, better cholesterol, all contributing to lowering that overall cardiovascular risk, which, remember, is the big killer in MASLD.
That's a really compelling outcome. But to really understand why it works so well, we need to go deeper, right, into the mechanism.
Yeah.
And then this is where it gets really interesting, involving a protein called PCK1.
Yes, exactly. This is probably the most novel part of the study from a biological perspective. They dug into how these drugs, particularly Telmisartan, were working in this early-stage fatty liver context.
Because Telmisartan was already known to do some good things in advanced MASLD.
Right. In later stages, where you have significant inflammation and fibrosis, or scarring, Telmisartan has shown some anti-inflammatory and anti-fibrotic effects.
Okay. Helps with the damage control later on.
Sort of, yeah. But this study was focused on the early stage, where the main problem is just the fat accumulation itself, before significant inflammation or scarring kicks in.
And what did they find Telmisartan was doing there?
Well, here, its fat-lowering effect wasn't primarily about inflammation. It seemed to be working by regulating this key metabolic protein, PCK1.
Okay, PCK1. Let's break that down. What does it do normally?
So, PCK1.
Yeah.
The full name is phosphoenolpyruvate carboxykinase 1.
Let's stick with PCK1.
Agrees. Yeah, PCK1 is fine. It's a really critical enzyme in the liver, a major gatekeeper in metabolism. Its main job typically is in gluconeogenesis.
Making new glucose, new sugar.
Exactly. The liver makes glucose from things that aren't carbs, like amino acids. PCK1 is central to controlling that process. Think of it as a master switch deciding where energy building blocks go.
Okay. And in the animals with MASLD, what happened to this PCK1 switch?
They found that in the livers of the animals on the high-fat, high-fructose diet, the amount of PCK1 protein was significantly reduced. The switch was turned down low.
And what effect did that have?
That seemed to push the liver's overall metabolism towards making and storing fat.
Okay, so low PCK1 equals more fat production. And Telmisartan.
Telmisartan treatment brought those reduced PCK1 levels back up to normal. It essentially turned the switch back on.
And turning that switch back on did more than just affect glucose.
Yes, this is the elegant part. Restoring PCK1 levels didn't just normalize glucose pathways, it actively reprogrammed the liver's internal metabolic factory.
How so? Can you give us an analogy, maybe?
Sure. Imagine the liver cell is a factory with two main assembly lines. One builds fat, lipids, for storage. The other builds glucose, sugar, mostly for energy elsewhere.
Okay, fat line and sugar line.
When PCK1 levels are restored by Telmisartan, it acts like a traffic director. It diverts the raw materials, the metabolic intermediates, away from the fat assembly line.
So it slows down fat production.
Significantly reduces lipid synthesis, yes. And it redirects those materials towards the glucose assembly line. It's essentially rerouting the internal workflow to favor making sugar over making fat.
Okay, but hang on. If you're pushing resources towards making more glucose inside the liver, doesn't that raise a red flag? Especially since MASLD often goes hand in hand with type 2 diabetes or insulin resistance. Are you just trading fatty liver for high blood sugar?
That is the absolutely critical question, and the researchers looked at this very carefully. They found that while, yes, the liver was producing more glucose internally because of the PCK1 boost, that extra glucose was not being excessively exported out into the bloodstream.
So it wasn't spilling out and raising blood sugar levels?
Exactly. It didn't seem to accumulate in the blood or worsen hyperglycemia. It appears to be more of a beneficial, localized metabolic shift within the liver cell, stopping it from making excess fat without causing a systemic blood sugar problem.
That's a really important distinction. A local fix, not a systemic issue.
Crucial, yeah.
So we have this combination approach. Lower doses, benefits for liver fat, blood pressure, cholesterol, and this newly understood PCK1 mechanism. Sounds incredibly promising. But we need to ground ourselves, right?
Yeah.
What's the big caveat here?
The big caveat, as always with this kind of research, is the translation from animals to humans. These are very strong results in well-established rat and zebrafish models, but they are still animal models.
So human trials are needed.
Absolutely. Clinical studies in actual MASLD patients are the essential next step to confirm that these benefits, the half-dose synergy, the PCK1 effect, the safety, all hold true in people.
Makes sense. What's next for the research team then? Where do they go from here?
Well, they have a clear path forward. One key thing is to test this combination in more advanced stages of MASLD.
When there's already scarring.
Exactly. When fibrosis is present, can the combination not just prevent fat buildup, but also help reverse existing damage? That's a huge question.
And presumably keep looking at the cardiovascular angle too.
Yes. They also plan to use more complex models that include both liver fibrosis and signs of cardiovascular disease, like atherosclerosis, hardening of the arteries.
To really prove that fixing the liver helps the heart in this context.
Precisely. To confirm that the liver benefits translate directly into reduced cardiovascular risk markers.
So stepping back, what's the real significance of this study? What should we take away?
I think the significance is multi-layered. It identifies a specific, potentially effective, and importantly, potentially safer combination therapy using drugs we already know well. That's huge, practically.
Right, the repurposing angle.
Yes. But it also provides a deeper understanding of the mechanism in early MASLD, highlighting this key role for PCK1 in regulating fat accumulation. That gives the whole field a new target to investigate for future therapies, maybe even beyond Telmisartan.
Okay, so let me try to recap the key takeaway for everyone listening. Researchers have found what looks like a powerful shortcut in the fight against fatty liver disease, MASLD, which affects so many people. By combining two existing drugs, one for lipids, one for blood pressure, they found they could use just half the normal dose of each and still effectively reverse liver fat in animal models.
That's the core finding.
And this combo not only helps the liver, but also tackles high blood pressure and cholesterol. Plus, they uncovered a new reason why it works, involving a protein called PCK1 that helps the liver reprogram itself to make less fat without dangerously raising blood sugar.
A really elegant piece of work combining clinical relevance with mechanistic insight.
So here's a final thought for you, our listener, to chew on. We've talked about this combination tackling liver fat, blood pressure, and lipids all at once.
Think about the potential ripple effect. If this approach works in humans, what could it mean for overall metabolic health? Maybe preventing or managing conditions often linked to MASLD, like type 2 diabetes. And thinking even bigger, if regulating this PCK1 protein is such a powerful metabolic switch in the liver, could targeting PCK1 become a strategy for other metabolic problems beyond just the liver? What other conditions might benefit from fine-tuning that crucial metabolic gatekeeper? Something to ponder.